Hello everyone and welcome to another edition of Nanotalks. This Nanotalk is a recording of another liquid phase electron microscopy user webinar given by Professor Nico Sommerdijk, who did fascinating research using our ocean system. In this webinar, he presents various research examples that illustrate how time-resolved TEM can be used to visualize the evolution of morphology and structure in solution from the sub-nanometer level up. Now we will switch to the recording. The presentation starts after a short introduction of the speaker. After the presentation, you can also listen to the Q&A with the audience. So I hope you enjoy this Nanotalk.
A very big welcome to our guest today, our speaker, Nico Sommerdijk, who I want to introduce to you with a few words. Nico Sommerdijk is a professor in the Department of Biochemistry at the Radboud University Radboud Institute for Molecular Life Sciences in the Netherlands. He obtained his PhD from the Radboud University Nijmegen in 1995. He has been a pioneer in the use of cryo-electron microscopy in soft matter, and more recently he became interested in liquid phase electron microscopy. In August last year, he moved to Radboud University Medical Center, Nijmegen, where he investigates the biochemistry and structure of collagen in both physiological and pathological situations. In particular, the mechanisms of assembly, cross-linking, and mineralization of collagen. He recently established the Radboud Electron Microscopy Center that supports the advanced electron microscopic analysis of biological materials. And that's, of course, where our systems come into play. And now I'm going to hand over the screen to our presenter of today, Nico.
Hello, ladies and gentlemen. Today, I am very happy to speak to you about how we are using liquid phase electron microscopy and the holders of DENSsolutions in my research. In my research, I'm interested in how inorganic materials are used in biological systems to create functional materials, like we see in bones, teeth, and seashells, where the organic assemblies are controlling the formation of inorganic materials. My research is now focusing on how bone is formed, and bone is formed by the mineralization of collagen. The mineralization of collagen with calcium phosphate. Collagen forms these fibrils in which calcium phosphate is present as these little crystals. And we have not only been interested in the details of these processes, but also in how we can understand what to do when these things are going wrong in patients. So far, we have been using cryo-electron microscopy predominantly because cryo-electron microscopy allows you to capture a process by very fast freezing. What you see here is a pair of tweezers holding a TEM grid. The TEM grid is then that has a droplet of the solution that you want to investigate. Now, using filtered paper, you can make that droplet into a very, very thin film that you can then subsequently shoot into liquid ethane. And that arrests the process. And you can do that at different time points. Now, we have been studying collagen mineralization, trying to get to the molecular details of that process. And what you then see is that you find the smallest calcium phosphate particles, little clusters that come together and infiltrate the collagen. You can monitor that process, and you can, apart from having information from TEM on the morphology, you can also combine that with structural information using electron diffraction, or even information on elemental composition using EDX. And if you want, you can even do that in 3D. So we have been able to understand quite a lot about these mineralization processes, and we have done similar studies for using different materials: calcium carbonate, calcium phosphate, as I showed, also organic crystals, iron oxides, and we can get to the smallest possible details. Well, we see here particles of maybe a nanometer in size. But what we don't get with cryo-electron microscopy is any information on either their past or their future, because by freezing the sample, we are losing all the dynamics. Now, after I had been working with cryo-electron microscopy, I had extensive discussions with my good friend Jim Diorio, who convinced me that using liquid phase electron microscopy could actually add the dynamics to my studies. And although we may not be able to get the same resolution in space, we will certainly get the same a better resolution in time. And that is basically how it started. Jim and I wrote this review paper where we are comparing what we can learn from liquid phase electron microscopy and from cryo-electron microscopy. And we are advocating there the use of the two techniques together to get complementary information. My interaction with Jim also led to the work of Paul Smeets, a joint PhD student that we had working both in his lab in the US and in my lab in the Netherlands. With Paul, we worked on investigation of the mechanisms underlying the formation of calcium phosphate in seashells. If we have a shell, we all know that we have a rough outside and a shiny inside. That on the shiny inside is nacre, is a aragonite, which is a specific form of calcium carbonate. And the aragonite is present as platelets that are stacked between organic layers. And these organic layers are the layers that are defining how the crystal will grow. They will define the size of the crystal, but also the crystallographic direction. And that is by the interaction with specific proteins and other biomolecules that are on the surface of these organic layers. And Paul's research was focusing on how certain glycoproteins could actually direct that process. The background of this study goes back quite a long time. In 1986 already, Leah Adadi and Steve Winder published a paper where they proposed that the sites where these calcium carbonate crystals in nacre would start to grow would combine two types of molecules. It would combine proteins that were rich in carboxylate groups that would direct the nucleation, and sugars rich in sulfates that would actually act as a sponge for the mineral, to bring the mineral together, and then it would then nucleate, directed by these proteins that are rich in carboxylate groups. Now, this is a theory that was never proven, and we thought that maybe we could actually show if it could indeed work in that way. And for that, we used liquid phase electron microscopy. First, Paul started to see if he could grow calcium carbonate crystals inside the holder inside the electron microscope. So we had a flow holder in which we could inject calcium ions into the cell, and we could also introduce carbonate ions into the cell. And if we were doing that, we could indeed see the growth of calcium carbonate particles, in this case vaterite, as we could see from the electron diffraction pattern. And we could see the calcium carbonate particle growing in less than minutes. And we could actually see that it starts growing from maybe 2-3 nanometers up to 20 nanometers in size. And our plan was to replace the sulfate-rich sugars that we find in biology by a sulfonated polystyrene, a polymeric a polymer with sulfonate groups that indeed that we could find that was interacting with calcium and actually acting as a calcium sponge. And when Paul introduced that polymer into the liquid cell, it forms complexes with the calcium, and it sits on the surface of the window of the cell. if we introduce it, we see these very vague blobs appearing. You can see here. And they are very vague because they consist of calcium and polymer, but mainly of water. These are actually hydrated globules that are sitting at the surface of the window. We can also visualize them with cryo-electron microscopy, and we see that indeed we have these round blobs in solution. So these are polymers containing calcium ions. And similar as in the biological situation where you have these sugars with sulfonate group that acts as calcium sponge, we now hoped that we could use these to nucleate calcium carbonate crystals. So we have these sponges with calcium ions sitting on the wall, and now we're introducing carbonate ion. And if we do that, we indeed see that only in these vague blobs, we see little particles of calcium carbonate growing. And we can only see them growing inside these blobs, and they stop at a certain point when basically all the calcium ions in the blob are consumed. And so we can monitor in time the growth of these particles. And then we come to the thing that we can find in liquid phase electron microscopy that we cannot find in other forms of electron microscopy. And that is that by putting all these pictures together in a timeline, we can actually work out the kinetics of the process. And we could see that the formation of amorphous calcium carbonate in the presence of these polymers follows a different timeline than the formation of veterite in the reaction without the polymers. From that information, we can get information about the supersaturation of the solution, and in fact, we can work out the mechanism by which these crystals form. Based on that information, we were able to propose that nature is using a so far undiscovered way of controlling crystallization. We all know that heterogeneous nucleation can speed up nucleation and can actually direct the nucleation of specific crystalline forms. But what we now found is that by making a sponge for the material, we can increase the local supersaturation and by that create new pathways for crystallization. And this is a way that, that nature uses apparently, which was so far not described. I now come to the second topic of my talk, in which we use liquid phase electron microscopy. And that is where we started to look if we were able to visualize organic materials, purely organic materials. And for that, of course, we need very careful considerations about the reaction conditions that we're using, but also about the electron dose that we are using and the way that we are recording our images. And the considerations about how to use liquid phase electron microscopy for soft matter and for biological materials, we have described together with the group of Niels de Jonge in this recent paper. Together with Niels de Jonge, we have been talking to DENSsolutions for already quite a number of years ago about the possibility to have a liquid holder, a liquid flow holder. And from these discussions, indeed, a holder was designed that we now know as the ocean holder. But when we were involved, it still had this name, the SLF30 FEI, because this was the microscope that we were using at the time. The holder has one inlet and one outlet, which allows us to do flow experiments. We have been using this holder and we have been very happy with it, because for us the big benefit was that it allows the assembly of the cells with a very high degree of success, and it also allows to control the bulging to make successfully or consistently holders that were thin enough to have good resolution. One of the experiments that we did was to look at the assembly of block copolymers, and specifically the assembly of block copolymers into vesicles. The block copolymer that we investigated was polyethylene oxide polycarbolacton, block copolymer that are used to form vesicles for biomedical applications, so for example for drug delivery purposes. And these vesicles are usually formed by the dissolution of the polymer in a good solvent for both blocks. In this case, we used acetone. And the block copolymer is an amphiphilic block copolymer. That means that this part, the hydrophilic part, dissolves well in water, and the hydrophobic part, the polycarbolacton, does not. So when we start in acetone, we have a homogeneous solution. But when we start adding water, we're starting to create a situation where this part of the block copolymer no longer wants to be dissolved. And in the end, these parts of the hydrophobic parts will all cluster together, forming the membrane of the vesicle. Now, for the mechanism of formation of these vesicles, there were two proposals in the literature. One was that it would start from a micelle. The micelle would grow big. It would grow so big that it would have a hollow interior, and from that a vesicle would form. The other mechanism in the literature was that we would start from the same micelle, that the micelle would grow out to a worm-like micelle, eventually to a flat membrane that then would close into the vesicle. And our aim was to investigate this process. And we wanted to do that in the ocean system, but there we had the challenge that we needed to mix two solutions while having only one inlet and one outlet. So to overcome this problem, we did the following. Starting with an empty cell, we entered the polymer solution in acetone, filling up the cell, as you can see here. And so we had polymer solution inside the membranes as well as around them. You can see here the filling of the membranes. And then we blow the solution away. We're blowing in air. We're blowing the solution away around the chip, so that only the solution in the chip would remain by capillary forces. And we can see that the chip still is filled. And in the last step, we introduce water surrounding the whole chip and diffusing in slowly from the side to change that acetone solution into a mixture of water and acetone in which the polymer vesicles could form. And this would now allow us to study if we have mechanism 1 going on or mechanism 2. And I will now and where you can see for yourself what is happening. What we see is that the vesicles are there from the beginning. The vesicles are there, and they become clearer and clearer and clearer as the process goes on. So what is happening? If we take all the 1,000 pictures that we made in time, and we take one of the particles and follow it back in time, we can make a cross-section of this particle and put all these cross-sections together. And if we do that, we see that the membrane of the vesicle is forming and it is becoming more and more prominent. We can compare with cryo-electron microscopy and see that we indeed in Cryo-TEM see the same type of vesicles, showing that the vesicle formation process is not affected by the continuous exposure to the electron. And we can open up the membrane, and we can straighten this membrane that you see here, and we do the same for the cryo, and we see that we have similar membrane types. We can now analyze the data a bit further, and we can look in detail, and we see that from the beginning we have a little blob that has not really a difference between the inside and the outside. And only in time we see that we get a membrane forming on the edge of the droplet. And here we see another way in which we depict what is happening. We see a droplet forming, and the droplet will get an enrichment of the membrane forming component at the edge of the droplet. So in the end, we can conclude that we have a dense liquid droplet in which the polymer, which is rich in polymer compared to the solution, and in which the polymer is actually assembling at the edge, forming micelles and linear micelles until the membrane is closed, and where we actually see that we have a combination of the two mechanisms. And it's not a micelle growing until we have forming an internal capsule and then forming a final vesicle. It's also not that we have membrane fragments that are then closing, but we have a unifying mechanism showing that both mechanisms in part were correct, but in fact were only forming two, let's say the two extremes of the process as it occurs in this system. Now, so we have shown that we can use liquid phase electron microscopy to study processes in soft materials, in soft materials assembly. Now, the next challenge for us is, can we actually look at biological materials with liquid phase electron microscopy? Well, we know from the literature that we can image biological materials, but now the trick is, can we actually, and the beauty would be if we could actually monitor processes. And monitoring biological processes, it has severe challenges. We know that enzymes stop to work already at 0.01 electrons per square angstrom. And maybe we can see something at this electron dose, but then still we want to make movies of these processes. And so far, if we look at the work of Niels de Jonge, Niels predicts that we can, at one electron, the total dose of 1 electron per square angstrom, we would be able to see a carbon-based object of about 5 nanometers. Now, if we take that into account, we may be able to reduce the dose that we need a little bit, but we also need more subsequent images. So we're looking to bridge a gap of about 100 to 1,000 times that we need to reduce the dose. So we are stuck with two questions. Right? Can we increase the information that we get out of every electron? And can we decrease the damage that we have that every electron does? And here we can benefit from the evolution of graphene liquid cells. We know that graphene liquid cells have appeared almost 10 years ago, where they would form very, very small pockets where we could do very high resolution imaging. But recently, it was found that these graphene cells do not just provide us with a very, very transparent window, they also help us to reduce the damage that the electron beam is causing by guiding away the excess electrons. And the problem thus far was only that it was very difficult to make these graphene cells. Now, in a collaboration with the group of Gregory Snyder in the University of Leiden in the Netherlands, we have been working on the encapsulation procedures. And together with them, we came up with a procedure where we could efficiently and effectively and reproducibly capture solutions inside a graphene liquid cell. And one of the most beautiful examples is the encapsulation of lipid vesicles that we show here, and with which we hope to do future fusion experiments. Now, ladies and gentlemen, I hope that I have shown you that we have, in my group, we have adopted liquid phase electron microscopy as a very important method to study processes with nanometer detail and with keeping the dynamics of the process. And I also hope to have made you realize that with the possibilities that we have now, liquid phase electron microscopy has grown into a technique that has attracted a wide community, all the way from material science to life science. And here we see the population, I would say, of the conference that was organized earlier this year by Niels de Jonge with my assistance, and which brought together this community in one dedicated conference. With that, I hope to have shown you that liquid phase electron microscopy is an important part of imaging in the future. And then leaves me only to thank the members of my group that have been contributing to the research that I presented, in particular Paul Smeets and Hang Long Wu, and my collaborators Jim de Jorio and Niels de Jong. I thank you very much for your attention.
All right, and I'd like to thank you for your presentation, and I think I can say that also for the audience. We still have around 50 people here with us who watch your presentation. So thank you very much for that, and also thank you for your kind words about our system, about the usability of our ocean system. So we still have a few minutes left. And I have two questions from the audience that we will now handle. The first question was from Idan Biran from the Weizmann Institute. Yes, the question is, hello, my question is to ask for Nico's opinion on liquid phase electron microscopy of small organic molecules. Is it possible? Is the question.
That is a very interesting question, and I'm expecting that what is the name of the person that asked the question?
Idan Biran from the Weizmann Institute.
I guess Idan works in maybe closely to Boris Repczynski. Who does wonderful work on cryo-electron microscopy on these assemblies of small molecules. If we go to this graph, we see that we have a limitation in our resolution that we can get from low contrast materials. We can hope to get a resolution of let's say of a nanometer, if we can bridge this factor 100 to 1000 from current state of the art. So we need really sensitive single electron detectors. We need phase plates, but we may also need very efficient acquisition techniques such as sparse imaging, as developed by Nigel Browning and now also by Niels de Jonge. And I guess that the use of graphene liquid cells will also make a big step in bridging that gap. Now, of course, small molecules are not active and are not relying on enzymatic action, but we need to assemble them to a certain size before we start seeing the aggregates they are forming. So in my view, yes, it is certainly possible, but we still need a couple of steps to be made in the methodology that we are developing.
Thank you, Nico.
All right, and thank you, Idan, for asking your question. The next question is from Hongyi Xu. Hi.
Can you hear me?
Yes,
I can hear you.
Great. Thanks, Nico, for the very nice talk.
And I'm from Stockholm University, so we work with microED. So my question was, for the vesicle study, how much electron dose did you put on these materials?
We used 30 electrons per, 0.3 electrons per square angstrom, as you can see here, per second.
Yeah. Okay, so that's the dose
rating. We had exposure times of 1 second, and we collected up to 1,000 images in per series. So that means, does that answer your question? Does that give you any insights?
Yes, it's great. So I was wondering, so if you use a very small electron dose, would you still be able to penetrate through and get signals afterwards? Yeah. Which I think 0.3 electron angstrom square per second is quite low. It's kind of the dose we use for studying protein crystals as well.
Okay, but I have to say that this is actually quite. We would use less if we can. For biological materials, this is on the high side. Certainly, if you want to, if you collect movies, you have to make multiple images, and so for every image, this dose is added. These images were recorded with, let's say, a relatively old camera. Not a very sensitive camera, and in a relatively old electron microscope. So with new technology, you could do the same with much less dose, or get far more resolution with the same electron dose.
Yes, exactly. Yeah, I understand. So with the direct detect electrons and powerful microscope, it's possible. And we also work with a very old microscope, and we work in diffraction mode. So with that, we can reduce the dose and increase the signal to noise ratio as well.
Yes.
Thanks for answering.
Just going to see if Peng-Han Lu is there, and if yes, I see the microphone is working. So welcome.
Yes, can you hear me?
Yes, we can.
Yes.
Thank you for the very nice talk. This is Peng-Han from UIsh. My question is about the dose budget. So there are some opinions in the community saying that the dose budget for liquid phase EM of biological imaging would be a little bit higher than the vitrified sample. I just wonder what's your opinion, Nico?
Yeah, so that is what we should. It goes two ways. If we have let's say some mild damage in the liquid phase, for example, by creating positive and negatively charged particles or charged fragments of a molecule, they can recombine. They can recombine if we give the system enough time. And in cryo-electron microscopy, when damage is done, damage is done. There's no way back. Because but we can, if we design the experiment such that we irradiate the sample and then give it a relaxation time and then do it again, and so we can actually have the sample relaxing from the stress that it got by the irradiation, and in that way we can actually prevent damage using the same dose as we would do in cryo-electron microscopy. So in that way, the dose budget in liquid phase electron microscopy for biological materials can be higher, indeed. But we have to take into account that in liquid phase electron microscopy, we want to make movies. And in cryo-electron microscopy, we in the end record stills. And so all the information goes into one image, and in a liquid phase electron microscopy, all the information goes into a movie of multiple images.
Yes, thank you.
I have Jordi von Seca, and I want to see if he's able to get to the microphone, perhaps. Jordi?
Yeah,
it's working. Yeah, hi.
Good night for the people there. I was wondering if once he has flow the wet air to empty the out part of the cell, the acetone lamella was kept there by a capillary phenomenon.
Yes, indeed. There's a very, very thin space, and it is kept there by capillary action.
So after that, you could enter the water.
Yes.
At the end,
so you could.
Yeah.
Can you still see me? Can you still see me?
Yeah, yeah.
Can you see my fingers? So when you enter, when the water enters, it will then diffuse into the cord compartment between the layers by diffusion. So it will diffuse into the acetone layer from all sides.
So in fact, you could combine any solution in principle that you want in this way.
Yes, yes, but we have to take into account the following. We did it with more different systems. It will take a while of diffusion before the new incoming solution actually reaches the viewing window, the viewing part of the chip. So the chip is much wider than its viewing window, and that means that the solution, when it, for example, it is a reaction, or it is a precipitation reaction, for example, that precipitation may already occur before the solution has reached the viewing window.
Exactly.
So you have to balance the concentrations and the reaction speeds to make sure that what you want to see is actually occurring in the center of your chip, so that you can actually have it in the viewing window.
Excellent. Thank you.
All right. Thank you very much for your question. I have another question here from Tanya Prozorov. She first, yes. She first thanks you for your presentation, and she asked what camera was used for image acquisition in your work with LPEM imaging of polymeric vesicles formation.
Hello, Tanya. Nice that you are here. The exact camera, the exact type of the camera, I don't know. It is the camera under the T20 that was bought in 2005. It's a 1K by 1K camera. So it's let's say it's a relatively low-tech camera, but I cannot recall the exact make. But it is so it's not nowhere near any fancy camera. It is really but it was the only microscope that we had available in which we could have a side entry holder, because our other microscope, as you know, was a Cryos and that has an autoloader and is not suitable for liquid phase electron microscopy.
All right. Tanya says, yeah, great, thank you. She was mostly wondering if it's not a 2K camera. So.
No, it is a 1K by 1K.
Yeah. We will now handle the last question from Barbara Malheiros from Brazil. And she asks, first of all, such a nice presentation. Very interesting work with liquid cell EM and beautiful applications. In this last project with the graphene that you've shown, how does it work experimentally to put the graphene sheets onto the chips? Does it have to be right before inserting the holder into the microscope, or can it be mounted and stored for a while?
this is a very, very good question. So we have been able to keep solutions inside the graphene liquid cells liquid and intact for 24 hours. But it is not a perfect technique yet, so it does not always allow us to keep the solution or the cell intact for that long. If you have a small leak somewhere, then you would like to go to the microscope earlier. But we are now trying to optimize the system such that we can actually encapsulate a sample and then take our time until we go to the microscope. So at this moment, it's somewhere between, let's say, half an hour and 24 hours.
All right, Barbara, thanks you for your answer. I want to thank you all for attending. I hope this was informative for you and that we, as is our aim, can be a platform for sharing knowledge in the worldwide in situ TM community. And then a big thanks to Nico. Nico, thank you very much for your clear presentation.
Okay, it was my pleasure. Thank you very much.
And that brings us to the end of this Nanotalk. To stay informed about upcoming webinars, I invite you to follow our social media channels, LinkedIn and Twitter. I also invite you to sign up for our monthly newsletter, in which we share interesting in situ research and technological innovations. I want to thank you for watching and hope to meet you at one of our next webinars.